Hi everyone, welcome again. I'm Wasim from Align Bio as a scientific developer.
I'm Erica, and this is the much requested troubleshooting guide. Um, as a person who is trained to do wet lab science in person, there's lots of things I pay attention to when I do experiments, and this is the tips and tricks for where that information lives when you're working in a cloud lab. You actually do get a ton of information about these experiments. And you need to know where it is so that you can look back at experiments and understand what happened and do troubleshooting. And so Wasim's going to walk through this. Um, Maybe one thing to know before we start is user code that's written gets compiled into unit operations, which are the individual steps that happen in order to execute a protocol. And the troubleshooting like some of this allows you to actually look at the individual unit operations, understand what happened in them, exactly what parameters were used. So sort of like the equivalent of looking back at your lab notebook and trying to reconstruct like what you did after an experiment. So, Wasim.
Great. Thank you, Erica, for that introduction. As she said, like there are, so whenever you provide an option in an experiment call, it gets compiled into an experiment protocol object, which contains all the information you need in order to execute the protocol. During the execution of the protocol, more information gets populated in that same object, as well as your data gets populated in the same object. So you can actually go back and look at all of these different information that has been populated throughout the execution of your protocol itself. And talking about protocols, there are two different types of protocols that can be like generated on Emerald's platform. I would kind of divide the protocols into two different categories. First would be a standalone type of protocol. I'll tell you about a little bit more about that in a second. The second would be the unit operation composed protocols, which Erica mentions here about, where if you specify different steps in order to perform an experiment, those steps get like binned into different unit operations that can be performed in the order you specify them in order to execute that protocol in lab. So first step, regardless of what protocol you are working with, is to look at the information that is contained within the protocol object. In order to do that, we use this quick little function called inspect. And when you run that, you get this wealth of information with a table in a tabular format, where you have all of these different parameters and other information being populated as the protocol was completed in lab. The fields that I especially want you to pay attention to is like the checkpoint progress, which kind of tells you what time different things were performed in lab. So for example, sample preparation was started at this time and ended at this time. Resources were gathered here, and then the assay plate was prepared, and then the thermocycling was performed because this is a PCR experiment, and then everything was returned, the materials was returned, and the experiment was completed. Similarly, other fields are like what sample volumes were used, what primers, what's the master mix, exact sample of the master mix that was used in this particular protocol. So if you suspect that there's an issue with this master mix, you can actually go back and trace it to where exactly it started off from, or from which sample it was like aliquoted out from. So there is the benefit of traceability, but you need to know how to exactly go back and look. And that's the purpose of this video, where you can actually see where all of this information is contained, so you can then go back and try to troubleshoot your experiment after it has been
And
completed.
and and half the battle is that there's actually so much information that's tracked that it can be even harder than if you're in person to
Right.
figure out what to look for and how to do detective work when you actually have such an enormous wealth of information about what happened.
Yep. Yeah. And then other important fields are like samples out, which basically tells you what sample is actually being generated during the execution of the protocol. For example, if you're running like a resuspend protocol where you're just resuspending the sample in place, there is no new sample or tube being generated. In that case, samples out is going to be like nothing. But if you are like transferring something from like your original container into like a new plate, there is a new plate with a new sample being created in such a case, there will be like a new samples out field, which will have all the sample IDs that are being generated during the execution of the protocol. Similarly, Emerald also captures all of the environmental data. That is the temperature of the room during the execution of the protocol, what was the relative humidity, in case your experiment is sensitive to that and you want to take a look at those data to kind of make an informed decision. And then here are like some unit operations that I was talking about that was performed in order to prepare the sample for PCR. And if you suspect that, oh, there might have been an issue with my sample preparation itself, you can actually take a look at these to see if there was any incorrect or irregular option that was being passed on to these individual unit operations. And as you can see, there was 20 microliters of the master mix, 2.5 of the primer, 2.5 of the reverse primer, and then all of these was kind of like combined with 25 microliters with the plate of the sample itself to generate like a 50 microliter total reaction volume. Also, you can also see what plate seal was used. And then additionally for PCR, these options can be different for each different types of protocol. For PCR, you have all the activation parameters, denaturation, primer annealing, and extension parameters that you want to take a look at in case you suspect that there might have been some foul or irregular thing happened during the execution of the protocol. Right. So now that we have kind of like an intro about how to look at an overview of information, let's look at the protocols that are composed of different unit operations. And for simplicity purposes, I'm taking a look at a simple protocol where I'm just labeling a plate and then transferring some liquid into that and then mixing that liquid and then finally covering the plate. So in order to see what different operations were performed for a particular robotic sample prep, and by robotic sample prep is usually a protocol that has been performed on a liquid handler. And in order to see what operations were performed, we're going to use this field called output unit operations, and it will give out these nice little graphics containing all of the information about individual unit operations that were performed during the execution of the protocol. Similarly, for manual sample preparation, where the lab operator has to perform these individual operations, you can still look at the output unit operations. And here, a container was labeled, and then like a small transfer was performed, and then finally the sample was labeled. So it's like a simple example that I'm showing you here. And for each of these individual transferred unit operations, you can actually expand it out. And if you click this plus icon, it'll give you all of the information about what the source of the sample was, what the destination was, how much of the source was transferred from the source to destination, if it was like handled in a liquid manner, how much percentage of the sample was transferred, what's the source well, source container itself, like what type of tips were used, like every information that you probably would need in order to debug is contained within that unit operation. So there is, like Erica mentioned, a wealth of information for you to go through, and you would have to know where exactly to look for. For operations or unit operations or experiments that are performed on a liquid handler, you can also additionally see something called these TADM pressure traces, which basically indicates, shows us the pressure that is being applied, or pressure traces of the liquid handler itself, the tip itself, to see if there was like a successful aspiration or a successful dispense from the tip. And you can take a look at those by using a quick function called plot TADM. And this function can be used on a whole protocol itself. I'm using it on a whole protocol here, object protocol robotic sample preparation. If I expand this out, you will see that it will have the pressure traces for each transfer unit operation performed. And here, in the first one here, it's saying that the source was like from the A1 well of this container to the B2 well of this plate. 3 microliters was transferred from here to here. And for the transfer, you can see there's a decent drop in pressure, which means there was a good aspiration, followed by a decent dispense. That means the transfer kind of was successful. Let's take a look at some of like irregular. So this, as you can see here, it looks like the the pressure is not very consistent, and this could indicate that there could be like a blockage in tip or something of that sort, and you probably would want to like investigate as to what happened in this plate on B2 location if later on in your experiment you noted that, hey, at B2 location here the data doesn't actually look right. So you have to probably go back and look at these pressure traces if all of your reagents were transferred successfully in there.
Yeah, so I mean, as a bench scientist, like you'll often like pipette and like look at it, and this is where that equivalent information lives in the cloud lab. So instead of like literally looking at the tube, you're looking at the sample from the perspective of the pipette tip,
Yeah.
as it sucks it up and dispenses it. Um, and so as Wasim said, this is similarly to if you're working in person, you may notice something's weird about a well in your plate, and then you would go look at the well, or in this case, look at the pressure traces for liquids going into that well to see if you think it was successful or not.
Yeah. Another way you could do is like if you perform like a large enough liquid transfer for it to change the volume significantly, you can actually perform experiment measure volume on like the initial container to again double confirm that some sample was transferred out of it.
Mm-hmm.
And that
Cool.
applies to experiments where you're performing it manually, where you do not have access to these pressure traces, because these are only accessible on like if things are performed on a liquid handler where the pressure is measured. But if it's performed like manually, for example, an operator is transferring 500 microliters from one Eppendorf tube to another one, there might not be a pressure trace for you to investigate. The best way to investigate would be to then measure the volume of the initial and final tube to make sure that there are enough volumes as expected.
Yeah.
Yeah, as you can see, some of these traces do not look great and worthwhile investigating a little bit more.
Mm-hmm.
Another cool little, let me see what I mean. So there is a field called for like individual samples. There is a field called sample history. It can be like a really big field or it can be like a really small field depending on like how many manipulations or transfers out, transfers in were performed on a particular sample, and you can basically get the history of the sample from the time it was generated. So for example, this sample was initialized in this robotic unit operation itself, and then other sample was transferred in, the experiment was started, some other sample was transferred out, some other sample was transferred out, another sample was transferred out, and so on. So you can basically trace to every different unit operation or different protocol where the sample was used or kind of basically it was involved. So
Mm-hmm.
it can be like a really big field depending on if like, for example, let's say you have like an original stock solution and everybody uses a stock solution every so often. And you can imagine like if it has been used in like 20, 30 different protocols, it will show us the history of every time it has been used in that protocol, and it could get like pretty overwhelming. But all of these are date stamped, so you can actually go back and look at that particular protocol if you suspect that there was an issue.
Cool.
All right. Yeah, like I said previously, the plot datum will give you all the traces of an experiment, but if you have narrowed down the problem to a particular unit operation, you can actually supply the plot datum function, the unit operation itself, to narrow down the number of traces that it's going to show. So for this, I'm just giving it the unit operation, and it's only going to show me the unit operation itself and the transfers that were performed within that particular unit operation. So again, these transfers are coming from the same unit operation. So there's that. Speaking of, there is another kind of like a specialty protocol that we have run into, which is the magnetic bead separation. And within the output unit operations, it will only show you the magnetic bead separation unit operation, which might not be super helpful if you want to investigate a little bit more. So what you want to do in that case is like copy that unit operation name, and then again within that unit operation there is a field called robotic unit operations, and that will give you a full list of individual unit operations that were performed within that particular unit operation. So there are like 3 different levels deep now.
Goodness.
Again, all of these unit operations are similar to the one that we have seen previously, label container, label sample. For the transfers, you can expand it and see exactly the pressure traces, dispense rates, like all of these options in case you want to like ever go back and take a look at those. For example, what tips were used. Let's say you're trying to transfer like 400 microliters, but accidentally you're using only like 150 microliter tip. So in such a case, you would not have a full transfer, even though the experiment might have been completed, because it did the pressure, it used the pressure to aspirate out, but it might have reached the capacity of the tip and might not have like transferred the full volume that you were intending. So
Won't the system not let you do that though?
there are certain cases that it might let you do, but like if you specifically give it the tip model, then it will kind of override the default settings.
Got it. Oh
Yeah.
wow. Okay.
And that's kind of like the downside of giving the user full control too, because then they kind of have to take responsibility of any kind of change that they make might not have been
Mm-hmm.
compatible with the system.
Yeah.
So yeah, as you can see, 3-level deep, all of these different unit operations were performed in order to accomplish the magnetic bead separation protocol.
And so, tell a story about what's a scenario in which you find yourself going back through these 3 levels deep.
So let's say there was a scenario where I was trying to purify DNA using Ampure beads, and I was doing it in like 4 replicates. And in the end, I saw that 3 of my replicates had the elution volume that I was expecting. However, one of them had like half the volume that I was expecting. So I had to go investigate the source of that well, and then try to see all of those different pressure traces to confirm that all of the liquid that I was expecting was transferred out of the magnetic bead solution and into the final plate or not. And even check if, sometimes I had to go back and trace it all the way up to sample preparation step where I'm adding the beads and the sample itself, because sometimes the bead can get stuck and there is a blockage on the tip if you specify the wrong kind of tip, and then beads might not have been transferred, and all you have in your preparation plate is your sample. So those are the
Mm-hmm.
things that you can go back and investigate
In
after.
the case you're referring to, what was the conclusion? What did you conclude about why your
Uh,
sample was?
so the conclusion was that the aspiration was being performed at a height that was like two
Ah.
millimeter above the bottom of the tube. Therefore,
Hmm.
not all of the sample from the sample where it was mixed with the beads was being eluted into the final preparation plate. So.
Got it. Wow.
Next, talking about standalone protocols. Again, the best way to get all of the information, like get an overview of all of the information, is to inspect that particular protocol object. Some of the fields that you might want to like keep a close look on is like samples in, which basically tells what samples are being taken into the protocol. These includes any reagents that need to be prepped in order to run the protocol, any buffers, any reagents in order to prep the instrument itself. All of these kind of include in samples in. And then for samples out, any samples that are being generated during the execution of the protocol. Sample preparation protocols, again, these are like kind of sub-protocols within the protocol itself that will tell you what operations were performed in order to prepare the sample for the experiment. Post-processing protocols are, these are some of the protocols that are performed after the completion of the experiment, and these are like measuring volume, imaging sample, measuring weight, etc., just to kind of get the post-experiment data for you to look at a later time point. Sometimes it might not be worthwhile to image the sample, because if you're trying to image the sample in like a plate, you have to remove the lid, and then it might lead to some sample evaporations and things of those sorts. So in case your samples are sensitive or like light sensitive, you might like refrain from like capturing images and things of those sort, things that you normally performed in labs. So you have to be much more vigilant and careful when specifying these options in like a platform like ECL.
Hmm.
So let's look at some of the cool fields that we can take a look in the agarose gel electrophoresis protocol. Again, we can look at these checkpoint progress to see what steps were performed at what different times. There was centrifuging performed of on the different samples. Again, I told you that there was a sample preparation protocol field, so there was a sub-protocol, operation protocol, object protocol centrifuge that was used to perform the centrifugation operation to prepare the samples itself. What samples, volumes were used, if there was any aliquoted samples. So aliquoted samples basically mean like if you have samples in like an original container, in order to run the experiment, it had to take the sample from the original container and move it to like a new container to be compatible for the experiment. Was that performed? Looks like, yeah, there were some samples that were being aliquoted out from their original containers to be used in the experiment. Here, there was a post-processing protocol of imaging samples, so after the experiment was completed, samples were being imaged. However, there is no measure volume, so samples was not being, the volume was not being measured. The reason being here, as we might have mentioned in a couple of our older videos, is that the ultrasonic measurement is not great when it comes to sample of low volumes, and here samples are like pretty low in volume, like less than 50 microliters, so that measurement would have been unreliable and probably not worth it to perform in this case.
Is there a way to just like globally disable measure volume for samples that are thought to be small volume? Or do you have
Unfortunately, there
to do it
isn't, so you have to
individually?
specify it for every single experiment, and there
Mm-hmm.
is same
Alas.
option name for it, so you just have to remember to say measure
Include
volume
that
goes to
everywhere.
false every single time, unfortunately.
Yep.
Because the default behavior is to measure all of these at the end of the experiment, unless
Mm-hmm.
otherwise specified.
Yep.
Data field is the important one, so this contains all the data that has been captured during the execution of the protocol. You can also look at the ladder data in case you want to take a look at those numbers to compare to your actual samples. You also have like the raw data file if you ever want to take a look at that. And this is like a zip file, and you would have to probably like expand it. Another important field that I was mentioning previously was the resolved options field. So these are the options that Emerald calculates on internally using their own logic after you specify the unresolved options. So these are the options that I specified for my experiment call, and the remaining options have been calculated automatically by Emerald. And in case you ever kind of feel that, hey, my sample doesn't look right, and there might have been like a parameter that should not have been used and could have led to this result, what you could do is like look at this resolved options field to see if the parameters that they have used after calculating it automatically were the right ones to be used for the experiment. And all of these were calculated on their own site.
What's an example of a time when you had to go back in here and you're
Right.
like, oh, that doesn't make sense. I would have set that differently. What's a good example?
So again, a good example is this exact one here. So loading dye volume I have not specified, and it calculated it to be 1.3 microliters. And again, as we know from our previous experience, transferring 1.3 microliters using
Too small.
is too small. So I later had to then look at this and then go back and modify it to be like 3 microliters at least of each
Mhm.
loading dye. And
Yep.
that led to good results. And as you can see from my data plotting, the results do not look amazing.
Yep.
So, so yeah, those are some of the cases in which you would have to go back and investigate. For
Hmm.
for gel electrophoresis type of protocols, you can also plot the gel animation, and this will basically give you the image that was captured as a video during the execution of the protocol. And all of these different like analysis functions are present in their in the catalog or the documentation that Emerald provides, and you can take a look at those, or you can go up top in the command center to commands analyze or plot to investigate more of those different functions that they provide.
Cool.
So as you can see, the ladders were run. Some of the samples do not look great. This lane practically looks empty, even though there is sample.
Mm-hmm.
So yeah, these are some of the ways you can go back and investigate after the protocol has been completed. And then you can, if you know what exactly the issue was, you can either rectify your parameters and run another protocol, or if even after investigating you're unable to figure out, you can always start like a support ticket, and Emerald would be happy to help out investigate as to what went wrong during the protocol. But this is
Cool.
a good first step for you to, again, delve into all of the data that is at your hand.
Yeah, I think it's also tough to understand the analogy between the data you have when you're in person and then where does that live when you're working in a cloud lab. So even just knowing that like pressure traces are available for you to look at after the experiment is run is just good to know. Cool. Thanks, Wasim.
Thank you, and hope you guys have fun working with Emerald.
Fun troubleshooting. Cool.